Hello everyone, welcome back. In today's video, we're diving into an important topic for your curriculum, electrophoresis. This technique plays a key role in instrumental methods of analysis and is widely used in pharmaceutical research and quality control. By the end of this video, you'll have a clear understanding of what electrophoresis is, how it works, the different types, and why it's so essential in the pharmaceutical industry. Let's jump right in. Electrophoresis is an analytical technique used to separate charged particles, like proteins or nucleic acids, based on their size and charge. This process is driven by an electric field, which causes molecules to migrate at different speeds, allowing us to separate them for analysis. In simple terms, when an electric field is applied, positively charged particles migrate towards the negative electrode, and negatively charged particles move toward the positive electrode. The rate at which these particles move depends on their size, shape, and charge. So, how does electrophoresis actually work? Let's take a closer look at the principle behind it. Electrophoresis relies on the fact that charged molecules move when placed in an electric field. The speed of migration depends on the particle's charge and size. Large molecules or particles with a high molecular weight move more slowly, while smaller molecules move faster. This principle allows us to separate mixtures of different substances based on their unique characteristics. Now, there are several types of electrophoresis methods, each suited for different types of molecules and applications. Let's go over the main ones you need to know for your course. First, there's paper electrophoresis. It uses a paper medium soaked in a buffer solution. It's a simple, affordable technique, often used for analyzing small molecules and amino acids. Then we have gel electrophoresis, which is the most common type. Here, molecules are separated in a gel matrix, like agarose or polyacrylamide. It's commonly used for DNA, RNA, and protein analysis. Gel electrophoresis offers great resolution for separating large biomolecules. Another important type is capillary electrophoresis, CE. This technique uses a narrow capillary tube and allows for very fast, precise separation with high resolution. It's often used in pharmaceutical and clinical research. Let's take a closer look at gel electrophoresis, as it's especially relevant for many pharmaceutical applications. In gel electrophoresis, a sample is loaded into wells at one end of a gel slab. When an electric field is applied, the molecules in the sample start migrating through the gel. Smaller molecules move faster, traveling further down the gel, while larger molecules lag behind. After a set period, the separated molecules form distinct bands in the gel, which can then be visualized using staining or UV light. This separation process is useful for identifying and analyzing proteins, nucleic acids, and other biomolecules. It's essential for DNA fingerprinting, protein purity analysis, and genetic studies. So, why is electrophoresis so important in the pharmaceutical industry? Here are a few key applications. Host, VO. In drug development, electrophoresis helps scientists understand protein binding, drug purity, and stability. It's also crucial for analyzing biosimilar drugs to ensure they match the original product. Host, VO. In clinical diagnostics, electrophoresis is used to identify proteins or genetic mutations, helping diagnose diseases like hemophilia, sickle cell anemia, and genetic disorders. Host, VO. And in quality control, electrophoresis ensures that biopharmaceuticals like vaccines and monoclonal antibodies meet purity and potency standards. Interpreting electrophoresis results is key to making the most of this technique. Here's a quick guide. Host, VO. In gel electrophoresis, results appear as bands. Each band represents a different molecule or fragment size. The distance each band travels in the gel corresponds to the molecule size. Smaller fragments travel further, while larger ones stay closer to the starting point. Host, VO. In capillary electrophoresis, results are displayed as peaks on a graph. The position and intensity of each peak help identify and quantify the molecules present in the sample. Here are a few tips to keep in mind if you're working with electrophoresis in the lab. 1. Always check the gel concentration. It should match the size range of your molecules. 2. Use fresh buffers to ensure consistent results. 3. Make sure the sample is properly loaded into the gel to avoid distorted bands. And that wraps up our introduction to electrophoresis. We covered the basics, types, and applications, as well as some practical tips. I hope this video helped you understand how this powerful analytical technique works and its importance in pharmaceutical analysis. Host, thanks for watching.